Hi, I'm Bree Ghostin. I'm from Ann Arbor, Michigan, and I have NF1.
And my name is William Raynard Golston. I'm from Detroit, Michigan. Diagnosed when I was around 9 years old and stuff. And then I didn't have that many tumors at that age, but then when I hit puberty, it just multiplied. Tumors on all of our bodies. It's on the nerves.
I was diagnosed when I was 5 months old, so they caught it pretty early. And in my case, my lesions are on the inside, so I have like brain tumors and them down my spine. And I was diagnosed with an optic nerve glioma when I was a kid, but unfortunately it grew as I got older. So I had to be on chemotherapy for 2.5 years to shrink the tumor. I don't have them on the outside as much, that's noticeable really. So I just try to tell people we just present differently, because every patient with NF, they have it differently. So no person's story is the same.
She's a strong person like I am. I'm very strong, I've been strong ever since I was young, because when I went to school, people used to tease me all the time. And so when I went swimming, they all get out the water and stuff, because they thought, I had something, but I didn't have anything that they could be fearful of. So it was hard growing up as a teenager, because I had, back when they didn't know what NF was back in the 60s. They did have a problem in school when I was young. So, but we didn't know that NF had affected me like that and stuff until I got older.
Even though my tumors aren't like as visible as his, I feel like we share that same bond because we both have this condition.
I hope to find a cure, not for me, but for all the people that's younger than me, because I'm older now, and they probably won't, find a cure for me, but the younger people that's coming up now.
I hope, more people know about it and it gets a larger platform. I also hope for a cure for it. So, and for it to be more researched and, have more funding. So in the next 10 years, hopefully there will be a cure or medication to help, slow the spread of tumors.
But I still live my life the best way I can.
Well, hello, everybody. I'm Bruce Korf. I'm a medical geneticist at University of Alabama at Birmingham, and here to talk about the NF Clinical Trials Consortium. I've had a long-standing interest in neurofibromatosis. Actually, in August, we'll celebrate the 40th anniversary of my involvement in NF, having set up a clinic back in August of 1983. But the focus today will be on the NF Clinical Trials Consortium, and in particular, there are 4 things that I want to try to cover in the short time that we have. The first is to explain why do we have a consortium? Why was it formed? What was the rationale? Then secondly, how does it work? Show you a little bit about the structure and the organization of the consortium. Then what have we done? And you'll see we've been operating for about 15 years, and a lot has been accomplished, and I'll try to give you a quick summary of that. And then finally, where are we going? We just were refunded in the last, now half year or so, for the next 10 years. So the consortium at the end of that funding period will have been operational for about 25 years. So I'll try to give you a window on where we see things going. All right, so the first point is why do we have a consortium in the first place? The dream in NF, and it was reflected in that video you just saw, is to develop treatments, obviously. And the pathway to developing a treatment is more or less illustrated here, which is that you begin on the left by trying to understand the basic biology. What is going on that causes tumors to form, or that causes learning disabilities, or that causes pain, or whatever manifestation you want to think about. Without understanding why these things happen, it's otherwise a kind of shot in the dark in terms of trying to develop a therapy. And a lot has been learned about NF1, NF2, schwannomatosis in the last several decades, mostly based on having identified the various genes that are involved. And you can't read it, I know, but the pathway that you're looking at on the left, that's the NF1 RAS signaling pathway. And once you identify how things function in the cell, and I wouldn't argue that we totally understand that now, but we do have a lot of information that we didn't, I can tell you when I started. Then comes the possibility of developing treatments. I show here the concept of drug discovery, and I'll say more about that in just a moment, but looking for small molecules as a kind of starting point that interfere with the function of that pathway, or better yet, set it straight, kind of fix what's not working when the pathway is perturbed because of a genetic change. If you find a promising drug, you're usually going to test it first in cell cultures, like neurofibromas that grow in culture, or schwannomas that grow in culture, to see if you can affect some kind of positive change in the behavior of those cells. And if you get to a point where you feel that's happening, you then are likely to go to an animal model. Mouse models, pretty typically, but not limited to that, are used to see if you can actually make tumors regress. If you have a model, and we do, for the various forms of NF, the ability to then see if you can shrink tumors, for example, or improve cognition, which you can test in a mouse. And then finally, if you have a drug that has passed through that, then the possibility of doing a clinical trial begins to be the case. Now, there are really, in theory, two sources of drugs. One is that you could basically invent one. You could use technologies that exist now, saying, well, here's the target. Now can we develop something that interferes with that target in some way or changes its behavior? Now, honestly, to do that is a very arduous task. It's said to cost $1 billion or so to go from essentially a blank slate to having a drug that you know that can be tested in people. Part of it is knowing it works. Part of it is knowing it's safe. The quicker approach is that there are existing drugs that have been developed for various reasons. And for NF1, for example, the RAS signaling pathway has been an area of interest in the pharmaceutical world for a long time because it's part of the kind of pathway that drives a lot of common cancers. So there was already a pretty good number of drugs that were developed for other reasons that you can repurpose, and that saves you a great deal. Because you don't have to start from scratch, and oftentimes there's already safety information that you know that this is going to be a drug that you could potentially use for treatment. Now, ultimately, you have to then go to clinical trials, and generally there are these 4 phases of clinical trials. Phase 1 is where you put a drug into trial where your goal is to see, is it safe? How does it get absorbed into the body? And how much of a dosage will people tolerate? What's the maximum tolerated dosage? Usually these are small trials just to get a kind of understanding of how people react to the drug, but you don't usually expect to see benefit in that kind of a situation. Phase 2 is where you actually test it on a larger number of people to see if it is working in the way you predict. Phase 3, you compare it with other treatments if they exist, and phase 4 would be after the drug has been approved by the FDA and is in use. You keep an eye on things to see if the drug is continuing to work and is safe. Most of the NF-related trials are phase 2 trials, or begin with phase 1 trials, perhaps, if the drug hasn't been tested in terms of dosage. But most of what we're focusing on is phase 2. It isn't usually even feasible to do a phase 3 trial in NF because we just don't have enough participants to make that possible, nor other treatments to compare to. Well, so in about 2004, I was actually the chair of the integration panel at the Congressionally Directed Medical Research Program in the Department of Defense, which, I don't know if you can read this, but there are lots of conditions nowadays that they are dedicated to funding research for, and I highlighted in red neurofibromatosis, which I think was one of the first back in the 90s, I believe, when this program first began. And so I was chair of what's called the integration panel back then, which makes the final funding decisions about whether a particular study should be funded or not. And one of the things that we noticed, and which frustrated us a great deal, was that we were already beginning to see proposals to do clinical trials, but they generally weren't possible to justify funding. And the reason why was they usually underestimated the complexity of doing a clinical trial. There are a lot of hoops you have to jump through to get a clinical trial. Obviously, recruiting participants, you have to get access to the study drugs, which oftentimes means making arrangements with pharmaceutical companies that have developed those drugs. There are significant hurdles in terms of getting regulatory approval. For example, the FDA. You need to get institutional review board approval, which can be formidable. And then once you start the trial, you have to monitor both compliance with the protocol and the quality of the data. And we were finding that most of the trials that were being proposed were underpowered, meaning they didn't have access to enough people to do a meaningful study, and they underestimated how much time and effort it would take to get through all of these different regulatory hurdles. And the question was, what can we do to deal with that? So back in around 2004, we convened a meeting. It was co-sponsored by the Department of Defense and by the National Institutes of Health. And it had a few dozen people who attended. And out of that came the idea of putting together a consortium which would have an operations center that would get sophisticated about how to handle those various regulatory hurdles, and a number of participant recruitment sites that had access to individuals who could potentially participate in trials, and that if you built this infrastructure, you could plug in different trials over time and have a much better chance of successfully completing them and coming to a firm conclusion. So within a year or so, the DoD put out an RFA, and it was really just to do a development of a consortium. It wasn't to actually do the consortium. And ultimately, UAB, which is where I am, was selected by the DoD review panel as the operations center. And at that time, 9 sites had competed for being patient recruitment sites. They got together and ultimately kind of defined how the consortium would work. And then from that, there was a competitive process to get funded by the DoD. So the first round of this was funded in 2007. It was a 5-year grant. It was then succeeded in 2012 by another 5-year grant. And in 2017, yet again. And then in 2022, the DoD, I think, had felt that this had been a good investment, but they also now were interested in looking at other funding mechanisms. And so the funding that now we have, and I'll say more about it later, is what DoD calls an other transaction award, which means they are very much involved in kind of the management of the consortium. They now sit on the steering committee, which I'll define in a minute. And we have funding now for 10 years, because one of the things we were finding was 5 years is a short time in clinical trial world in terms of developing a trial and then recruiting people to participate in analyzing the data. The time flies by faster than you could imagine. All right, so how does it function? Well, so the goals of the consortium defined here is to develop innovative biologically based therapies for children and adults with neurofibromatosis, defined broadly, NF1, NF2, schwannomatosis, and to implement hypothesis-driven, sort of scientifically informed translational trials for multiple manifestations of NF, hoping to improve the outcome for children and adults. Now, the original award, which was defined by the DoD, was 9 sites. I think those are shown in red here. And then in 2011, we added 4 new members, and then came up with the notion of the affiliate site. And what that means is many of the sites in the original group were children's hospitals, because that's where a lot of the expertise and interest in NF had started, which was fine, but they weren't in a good position to recruit adults, and we did want adults to be included in trials. And so those that were at children's hospitals affiliated with sites that had access to the facilities to manage adult individuals with NF. So that was the kind of origin of the affiliate sites. Then in 2017, 2 new sites and 2 new affiliates were added. And we've also had a few ad hoc sites that were not part of the consortium, but came to us with ideas to develop protocols. And when they did, they were also kind of incorporated as a site to get that protocol kind of completed. We do have a site evaluation process. It is a very significant investment of time and energy that the sites need to put in, and there are high expectations for their performance. And so I won't go into great detail here except to say we do monitor metrics. How many people have they enrolled into trials? How active have they been in terms of helping to formulate protocols and participate in various of the scientific activities of the consortium? And there are reviews, and actually there have been sites that ultimately were dropped because they didn't pass muster in terms of the site evaluation, and that has made room for adding other sites over time. So there are some frequently asked questions, and I know from looking at the chat that had preceded the meeting that these are things that are on many of your minds. If you look at the map of the country, which we have a booth outside where the map is shown, I didn't bring it in as a slide, there's a big swath of the middle of the country that's pretty empty in terms of recruitment sites, and you may ask, so why is that? So the reason that there aren't more sites, honestly, is as much as anything else a combination of budget and I guess you could call it bandwidth. What I mean by that is the budget for this sounds like a lot of money, but when it comes to doing clinical trials, they are very expensive to do. And the sites get a small amount of money, but they will tell you that they lose money on every trial that they perform because the time and effort that it takes for them to do the work is not completely compensated by the funding that we have. So we can't add new sites without even further reducing that to a point where there's almost nothing on a given site. So we have a kind of fixed number of sites based on the fact that our budget is finite and we can't really increase it. Now you probably have heard that there are instances of clinical trial organizations that pharmaceutical companies work with where they go out and they find places all over the country, including very often private practices, and get them to do trials. The problem is it's a monumentally expensive undertaking compared to what we're able to do. And usually you don't get very many participants per site when you do that. So it's just not a feasible option budgetarily, plus the effort that goes into launching a site, all the regulatory things that have to be done. Our operations center literally doesn't have the bandwidth to handle 100 sites, which would be great if we could, but the time and effort it takes to do that is really enormous. So it's just been very difficult. And finally, there's the question of, well, we don't control who volunteers to be a site. When we put out an RFA, people will respond, and we look for individual sites that have access to enough people with NF to be able to recruit and have experience in doing clinical trials. And also the consortium is not just a place that recruits people. If a pharmaceutical company decides to do a trial, they can pay an organization to do all the logistics. But we're doing a lot more than that because we're actually conceiving these trials, things that the companies might not think to do on their own, but which our faculty are interested in. And we will bring companies to the table that otherwise probably wouldn't have focused on this. But that means our sites are scientifically involved, not just doing recruitment. So I realize that the challenge that many face with this. And to be honest, at the end of the day, what's most important is getting these trials done as quickly, safely, and efficiently as possible, leading to an answer. Of course, everybody benefits in the long run if we come up with an answer, hopefully one that indicates that there's activity in a particular drug. Now, another question is, so how do sites get selected? So each grant cycle, we have issued a RFA. Usually there'll be a few sites that we feel haven't performed up to what we expect, and we will then have them compete with other sites to be parts of the consortium, and that is likely to happen in the near future for this grant cycle. Many people ask, well, I don't live near a site, but can I participate anyway? And the answer is sort of a yes and no, I guess, and that is yes, you can. And we've had some people who have traveled. The problem is we cannot have a site locally be your kind of anchor point for involvement. You have to come to a site that's part of the consortium if you're going to actually be a participant in a trial. Now, there may be some simple things, some blood tests, just routine safety monitoring, for example, that can be done remotely. But the major evaluations have to be done at a consortium site. They are complicated, and they require sometimes special equipment and a lot of experience and knowledge, and it isn't possible to do that remotely. And I'll say during the pandemic, there was a relaxation of those rules to a degree because it wasn't safe for people to travel. But the rules, and it's not rules we make, they're much more global rules, including with the FDA and the institutional review boards, now do require you to come to a site. And we don't have travel funds. The budget just doesn't allow for that. So there have been a few people who have done that. And we've had people, even from outside the US, have inquired. The other thing to know is that the budgets do not cover all of the tests that might need to be done. Imaging studies frequently, for example, are billed to insurance because they're part of standard care. And if a person comes from outside the country and they don't have US health insurance, they may very well not be able to afford the payment for the imaging studies and other things. So yes, it's theoretically possible, that pragmatically it's not so easy. All right, well, here's an org chart of the consortium, and I think the writing is too small to see most of it. It maybe will impress you. It's a pretty complicated structure. I'll just tell you, on the far left is the operations center. That's at UAB. And basically, we do all the logistics and regulatory activities. UAB now is the single IRB, that is the ethics review board for the entire consortium. We interact with the Food and Drug Administration because most of our trials require an investigational new drug application, which is a very tightly regulated kind of process. And we have staff who are very good at kind of navigating that. And then we oftentimes become the IND holder for a particular trial. We disseminate all the information about the protocols to the sites, which are now, I think, 24, 25 sites, and manage all of the kind of regulatory reporting of adverse events that occur in the context of treatment and so on. What's off to the right is the data center, which actually had been at UAB, but we transferred it to Children's Hospital of Philadelphia this year with the new grant cycle, largely because we felt they had better infrastructure for handling these kinds of trials, especially if there's significant FDA involvement. That's something that they were very well set up to do, so we made the decision to move the data management. And then in the middle, the two groups have to interact because part of what goes on at UAB with the operations has to interact with the data center, so there's a bit of overlap between the two. This too is probably unreadable, but it once again impresses you, I hope, with just how much activity there has been in the consortium. These are the committees. These are committees that focus on particular condition manifestations. For example, there's a neurofibroma committee, there's a committee for schwannomatosis, for NF2. There is a quality of life committee that helps to define measures to assess whether quality of life has improved as a consequence of treatment, not just as a, let's say, a tumor shrunk in size. Radiology committee helped to define some of the radiology approaches that are used. There's a biology committee that looks at the underlying scientific rationale for a clinical trial. And these committees meet on a regular basis, and they are the source of the protocols that we develop. So again, the consortium members are not only recruiting individuals to trials, but also very much involved scientifically to develop trials that would otherwise almost certainly never happen. So what's been accomplished in the now 15 years that we've had a consortium? Well, here's just some numbers to give you an idea of some of the milestones. There have been 15 clinical trials over this time with just shy of 500 participants with NF2 and NF1, but we haven't yet had a trial in schwannomatosis. Come back to that point in a second. More than 20 papers published in the peer-reviewed literature, more than 30 abstracts, that is presentations at international meetings, including the NF conference, more than 40 invited talks, and then we have leveraged more than $5 million in funding, largely from pharmaceutical companies, but also from donors, and Children's Tumor Foundation has been supportive of activities as well. So what we get from the DoD is only the start, and we've needed to be able to bring others to the table because, as I said before, these are really expensive trials to make happen. So this is showing the trials over the first 3 iterations up to the current one, but at the upper right you can see things that are in development currently. And I'm not going to try to read them all to you, and I'll show you a few sort of selected examples of trials that have been completed by the consortium. Above the line, those are trials that are done completely by the consortium. Below the line, those are collaborative trials. We've particularly collaborated with the Sarcoma Alliance for malignant manifestations of NF1, malignant peripheral nerve sheath tumor, SARC, as they call themselves, has an infrastructure for doing clinical trials for sarcomas, which is the kind of tumor a malignant peripheral nerve sheath tumor is. And we've collaborated with them so as to try to do this as efficiently as possible. In gray are NF1 related trials, and there are 2 trials you see in green. These are NF2 related trials. Obvious questions are, why more NF1 than NF2? And the second question is, why no schwannomatosis? So part of the answer to that, I think, is simply that more is known about the underlying biology of NF1, so that it was in many ways easier to identify candidate treatments for NF1, and many of them were kind of ready to go, and that made it much easier to get a trial underway. There have been a couple of NF2 related trials, but it has been more challenging to identify the candidates, and that's even been harder yet in schwannomatosis. We had one trial that was moving along. It was really focused on pain management in schwannomatosis. At the end of the day, it was in conjunction with the pharmaceutical company, and the way they wanted to structure the trial, it just wasn't possible to mesh that with the way the consortium is organized. But the schwannomatosis committee is continuing to try to develop clinical trials for that entity as well. We do have, and it's clustered at the very top, a number of trials that are in development. In fact, the consortium met just last week in a day-long Zoom meeting to go over the status of the trials, and they're at various stages of regulatory approval at this point. We have an external advisory committee that is separate from the consortium and does a scientific review of every protocol so that there's no sort of conflict between people who have developed it. We have people who are separate from the consortium who really scrutinize it and criticize it and ultimately make suggestions for how to improve it, the outcome being, I believe, a much better trial than it would have been if it had not gone through that kind of peer review. So there's that, there's FDA approval and ultimately IRB approval that is taking place. There was one trial that we were pretty far along with, and it was a sarcoma trial, and then the pharmaceutical company that was proposing to, or was arranged to provide one of the drugs, decided to stop developing that drug, and so that kind of temporarily put a halt to the ability to do that trial. Luckily, we've identified an alternative drug now, and so that trial once again is moving forward. But these are the kind of things that we have to kind of deal with, that it is a moving target in this world of drug development. Just give you a window on a few of the trials that have been done. One of the first trials, I think it actually was the first trial, was called sirolimus treatment of plexiform neurofibromas. It went by the acronym STOP-N. And you're looking at 2 different displays of data, and the one on the right is probably the one mostly to focus on. It's called a waterfall plot, maybe for obvious reasons. And what it shows is each bar, each vertical bar, is a different person who was treated on that trial. And The y-axis is essentially the change in volume of the tumor. These were plexiform neurofibromas measured by volumetric MRI. And so the idea here is that if the tumor grew on treatment, the bar goes up, and if the tumor shrank on treatment, the bar goes down. And you can see to the right, there was a cluster of tumors that did reduce in volume. We have tended to use a 20% reduction in volume as a kind of cutoff for what we view as a successful trial, because short of that, although it looks like maybe you're doing some good, there's a real risk of being misled, because the precision of the measurement is not that enormous, and you could actually be within measurement error. Plus, it's not clear that a 10% reduction in volume makes any difference clinically, that a person would even notice that. So we've tended to use 20% as a kind of cutoff. And what we found here was, there was some evidence of activity, but it wasn't enough to feel that this was a drug that had a future in treatment of plexiform neurofibromas, at least not as a standalone treatment. Sirolimus, by the way, is a drug that actually doesn't directly interfere with the RAS signaling pathway, but there was some evidence that the target of sirolimus, which is called mTOR, it's just a protein in the cell, is also hyperactive in NF in neurofibromas, and that was the rationale. Sirolimus actually is a drug that's used in treatment of trying to suppress the immune system for transplant recipients. So it was a pretty widely used medication. It just, at the end of the day, we didn't feel there was enough evidence to feel that it was a successful trial. Everolimus is a similar drug, actually, and this is looking at low-grade gliomas, things like optic gliomas. And what you're looking at here is every time somebody develops a recurrence of tumor, the curve kind of pops down a notch. And we found ultimately that vision did improve in a few instances. It stayed stable, and these were all people with progressive tumors at the outset in a good number, and a few progressed in spite of treatment. So it was a somewhat more encouraging trial outcome. So NF104 was a NF2 trial. It was the first NF2 trial that we did. And I'm showing you, by the way, all published data, and so the screenshots to the right are the references of where this was published. And bevacizumab is, it's also known as Avastin. You might have heard of it by its trade name. It was used to treat vestibular schwannomas. Scott Plotkin at MGH in Boston had shown that bevacizumab seemed to be effective. This was a larger trial. And sure enough, I mean, there were a substantial number of people. You want to see as many as possible under the curve going down rather than to the left going up. And yes, there was a pretty good response in that trial. Then NF105 was another plexiform neurofibroma trial using the drug cabozantinib. Cabozantinib interferes with the cell membrane receptor that activates RAS being the kind of master switch that is hyperactive when NF1 is missing, because NF1 tends to essentially turn the switch off. And the way the switch gets turned on in the first place is various things bind to the cell surface that are signals telling the cell to grow. And ordinarily, NF1 would kind of terminate that, so the cell stops growing. When NF1 is missing, the cell doesn't get that signal, keeps growing. Cabozantinib interferes with the signal at the cell surface. And you can see the waterfall plot was pretty encouraging. Most of the individuals went down in terms of tumor volume, and there were a fair number who did cross the 20% line. Now, there were side effects from this, and these were adults treated with cabozantinib, but it was considered to be an encouraging outcome, and it is going to be a drug that will continue to be studied in future trials by the consortium. Now, the trial that ultimately resulted in the approval of selumetinib, that I'm sure most of you are familiar with, that was not a consortium trial. That was done at the National Cancer Institute and as a phase 1 trial, and then they recruited just a few other sites, but it was totally run by the National Cancer Institute. But there are many other MEK inhibitors. These are inhibitors of some of the, or at least one of the proteins that is part of the kind of cascade from RAS ultimately to the cell nucleus. And so selumetinib had been encouraging, but there are many other MEK inhibitors, as they're called, and mirdametinib is one of them. And so NF-106 was looking at that drug. In the same story, there were more people whose tumors shrank than whose tumors progressed. And there were a fair number who exceeded that 20% reduction line, once again, indicating a positive outcome. It's a little difficult, I should say, to compare this with the selumetinib trial because some of the things that were done were a little bit different. The age range in particular who were treated were different. So I don't think we have data that tells you one drug is better than another at this point. That kind of study has not been done. But at least there was some positive signal here. So what have we learned in the now 15 years since the consortium was launched? First thing is, trials differ in terms of their focus. We've had trials that have enrolled very quickly, and I think most of the plexiform neurofibroma trials and the vestibular schwannoma trials, for that matter too, have been in that category. 'Cause honestly, there's a lot of people who are dealing with those kinds of complications and are hoping to see treatments. And so usually those trials have enrolled fairly quickly. Some trials have been more challenging to enroll to. For example, there was one done early on looking at learning disabilities. There was some evidence to suggest that lovastatin, which is used in treating hypercholesterolemia, perhaps would improve cognition, and that was based on some mouse data. The outcome was we couldn't really find good evidence that it was effective, but it took a long time to enroll, and part of the reason was I think people were much more motivated to enroll in trials or to have their children enroll in trials if they were dealing with something that was medically very significant, like airway compression from a plexiform neurofibroma, where you had to do something. The question is what? And on the other hand, having otherwise healthy children go on a medication hoping to improve cognition, that was a much harder thing to ask people to do. And we've had only one trial in the 15 or so that we ultimately couldn't complete, and that was a trial looking at bone dysplasia, the tibial dysplasia that sometimes occurs in NF1, using a treatment that the problem was many orthopedists were using it anyway, and it had never really been proved to be effective. And that was the point of the trial, was to determine if it was. But the problem was a lot of orthopedists had already decided that they just wanted to use it. So getting people to randomize and agree to either be using it or not using it was really difficult. And ultimately, we just couldn't enroll enough people to do that. So at this point, it's sort of a faith-based treatment in a way. There's good rationale for it. I don't think we have complete understanding of whether it truly makes a difference, but it wasn't possible to convince people to do a trial. So, I mean, we've learned a lot of lessons there. The sites differ too in how well they can complete trials. There are some sites, mostly the ones that are dealing with oncology-related problems every day, that are very robust and have been very successful. There are other sites have less experience. Some of those don't manage to stay in the consortium or even choose to. And so it really takes a site with a lot of sophistication how to run a clinical trial. It is complicated, and as I told you before, they lose money on these trials, and so the institution is kind of footing the bill, and that means only some sites are going to really be committed to doing this. We certainly learned a lot about how to contract with pharmaceutical industries and with the sites. We have, at this point, about 24 sites, I think, 25 counting UAB, and every one of them has to have a separate contract for every trial. So when you do the arithmetic, it adds up in a hurry. And we've got people, including grant specialists and attorneys, that are working pretty much full time on just grinding these contracts through. And it's one of the things that makes it hard to expand the consortium because the amount of time and effort. And you make a particular deal with a pharmaceutical company because that's the way they're insisting that things go. And some site says, well, we don't like that clause in the contract, and we don't really have the power to change that. So these are negotiations that are not simple to do. In the early days of the consortium, every human subjects research project of any kind has to be approved by an institutional review board who monitor the ethics, really, of how the study, whatever it might be, is conducted, and not only how it originally designed, but they also monitor its ongoing compliance with the protocol. Each institution participating has its own IRB, and they have a somewhat regional kind of flavor, and certain IRBs might be more willing to allow certain things to go on and maybe others not. And that creates sort of a Tower of Babel in a way when you're trying to do a trial, because that means you have to negotiate with 24 different IRBs, which is an enormously complicated undertaking. The last iteration of the consortium, not the current one, but the one from 2017, we finally were able to eliminate that as a challenge. And I think part of it was because the entire research world had come to realize that that was a cumbersome mechanism. And so now these multi-center studies, whatever they may be, including clinical trials groups, tend to use a single IRB, which serves as the IRB for the entire project. And the local IRBs agree to trust the single IRB to make the decisions on their behalf. The consent forms get customized to the institutional requirements, but they cede control, and so that's very substantially simplified the process. Although it's not a simple one, even with that, it usually goes through multiple iterations to satisfy the IRB. And then the Department of Defense, who are the funders here, they have their own IRB. And so we do have to get their approval of everything as well. But we've made a lot of progress here because that was a bottleneck, and it's much less so now. And a point I made earlier, in which I'll just reiterate, that the sites are not adequately funded. They all lose money in doing this work. And the reason they do it is because they're committed to it, not because it's a money-making operation for them. And we're trying as best we can to try to capture exactly what the true costs are. And I'll show you in a minute why that's important. But it is really as much a labor of love as it is anything else that keeps these sites that are working with us. So where are we headed now in this next 10 years? So the new other transaction award mechanism has started. And one of the things that was the case before, which is no longer the case, is I told you there was an external review committee that does a scientific review of all the protocols. There was also an external advisory committee, which advised the DoD in terms of just how things were going. And it actually created a lot of kind of challenges because we were essentially answering to two different external groups. And you could sort of go ping pong between the two, trying to keep them all happy. Now that's been eliminated. So there's one external scientific review, and the DoD actually sits on the steering committee, which consists of the principal investigators of all the participating sites. And they get to vote ultimately on which trials are going to go forward. We are interested in expanding areas of focus. I told you earlier, it's been very much on plexiform neurofibromas, some learning disability related projects, vestibular schwannomas, and sarcomas. And we know there are lots of other manifestations of NF. There is now in consideration another bone-related trial. There's an attention deficit disorder-related trial. And there are additional NF2-related trials. So there's a lot of interest in trying to expand our focus into other manifestations of the various forms of NF. We're now also looking into creating a kind of registry because, when you look at individuals with plexiform neurofibromas, if one thing doesn't work, maybe another thing will. And if we have a better kind of central registry of who's affected with different manifestations and interested in participating in trials, it allows us to kind of plug into different trials more efficiently. Imaging, MRI, for example, has been really critical, because that's how we know if tumors shrink or not. And we're trying to build a more kind of standardized image repository. And we don't necessarily always obtain tissue in the process of doing a clinical trial, especially like for plexiform neurofibromas. It isn't standard practice to biopsy those, because it's pretty obvious what they are. But sometimes tissue does become available, and we're trying to develop a much more kind of established tissue bank to facilitate scientific studies. Patient advocates have been involved in the oversight, less so in the development of trials, and that's something we're interested in including the sort of patient advocate voice. If you look at the diversity of participants by race and ethnicity and geography, it's not reflective of the country. And we're trying to work with our sites to try to improve the representation of diverse populations. We've been working a lot to build a group of young investigators, because many people, myself and maybe chief among them, are not getting any younger, and we need to see other people kind of come in and get involved. I think there's been some pretty good success in having young investigators get involved as what's really the next generation. And finally, we actually are obligated by the DoD to raise matching funds. So there's a $25 million award as this OTA over 10 years. And the expectation is we'll raise that much again. And that's, I know it sounds like a lot of money. It isn't when you start looking at what it costs to do these trials. And it's actually a pittance compared to some big pharmaceutical company sponsored trials. We are expected to raise matching funds. Much of it will probably come from pharmaceutical involvement, who are sponsoring, for example, the acquisition or the ability of us to get access to drug. They help to pay for some ancillary studies and many other things. It's part of it. It's not limited to that. CTF has been generous in helping us with some needs. We've had some philanthropic donations. And we're trying to account for the amount of time and effort our sites put in. I told you they lose money. Now we're trying to figure out how much, because in a way that's in-kind support that institutions are providing. All right, so I'm going to conclude now. I think there's about 5 minutes left for questions. Just want to point out, here's a picture taken actually something over a year ago at one of the consortium meetings. We meet as a group. Nowadays, it's once a year by Zoom and once a year in person. This was obviously one of the in-person meetings. And just to acknowledge the CDMRP, the Department of Defense, who have been supporting us now for 15 plus years. The many staff of the Operations Center, one of whom is here at this meeting, and come and visit the table that she has set up outside. Our many participant recruitment sites who were mentioned earlier. Our pharma partners, but then especially also the 480 plus participants who have actually worked with us to participate in these clinical trials. So with that, thank you for this opportunity, and I think just a few minutes for questions.
Do we have any questions from the audience? Oh, great. I'm coming to you.
Is there one collective site to see which trials are currently going on, instead of getting like different information at different times?
I'm sorry, it wasn't.
Is there one collective site to see all the trials that might be going on?
Oh, there's one place you can look at?
And see what's currently?
Yeah, I think we do have a website for the consortium, which should list those. Whether it's been totally updated, I can't tell you off the top of my head, but there is, and the DoD also has information about the consortium and the various trials that have been done. The other place, by the way, hopefully you're familiar with this, is clinicaltrials.gov. This is a federal site, and it tracks all clinical trials, literally all clinical trials for everything, including NF. And we're required to register our trials with clinicaltrials.gov. That includes not only the kind of way the trial is designed and who and where are recruiting, but also once the trial is done, we're obligated to put the outcomes there. So actually, that's probably the single best place to look in general.
Maybe one more question. Great. Hi, my husband Derek has NF2, and I was just wondering if you have any tips for the hoops to be a participant in a trial, being the middle woman for authorization from his local physicians, the site, his insurance feels like a full-time job. And so I've been trying to see if he could be in a trial for over a year. Just wondering if you have any tips for those hoops on our end.
Yeah, obviously it starts with defining the trial that you're interested in and making contact with the coordinator at the site that's actually doing the trial. The first thing you want to know is he eligible for the trial? And each of these trials has strict eligibility criteria, which are really adhered to, like, cast in stone. There's absolutely no flexibility. If it says it has to be 1 millimeter and it's 0.99 millimeters, it won't be possible to come on, for example. So that's the first thing, is you want to know that person is eligible to participate to begin with. If they are, then you have to understand what it's going to take in terms of whether you have to travel, whether there are things that insurance has to cover. Most of the time, the sites will work with you to define what your insurance company will permit, because each insurance company is idiosyncratic in terms of what they do and don't cover. And you certainly want to get pre-authorization for any studies that are not covered by the trial, because you don't want to be surprised by a substantial bill. But most of the time, if you're eligible and willing to travel to the site, then they'll work with you and your insurance company to figure out what they will and won't cover.